Alrighty, so I'll be talking to you today about whole brain transcriptomic axes of human brain organization. So as we all know, brain function is distributed in space. Obviously, a central theme of this conference is mapping brain function, and a great deal of effort goes into finding ways to divide the brain into regions with unique physical or functional properties. But I think something that we take for granted is the fact that brain function is organized as a spatial topography, where distal regions of the brain contribute quite distinctly to behavior. And one of the reasons for this is likely related to how distinct brain regions or areas form and develop in the first place, a process called regionalization or arealization. So imagine we have some sort of neural compartment present in very early phase of brain development. And in one part of the compartment, we have a source where some protein, maybe a morphogen of some sort, is being produced. The protein source will diffuse into the compartment, leading to a protein concentration gradient, partially constrained by the dimensions of the compartment itself. And we know that the concentration gradients like this help to recruit proteins that are differentially attracted to or repelled by the presence of our original protein to different parts of the compartment. So then let's say, another protein gets recruited to a different part of the compartment and it too begins to diffuse, and this will in turn cause a more complex gradient. And once again, proteins will be differentially attracted to or repelled by different parts of the neural compartment, depending on the relationship with those initial molecular sources. And as this process continues, the parts of the compartment begin to become more distinct as their molecular features become more unique through this process of differential recruitment. And before we know it, we can define distinct areas or regions of the compartment that, by way of hosting different proteins, may become functionally distinct throughout the developmental process. So this process of regionalization has been quite well described in simpler models, for example, within the spinal cord, as you see here on the left, or in the simple model animals like Drosophila on the right. And in both cases here, you can see different morphogens creating expression gradients, which help to recruit other proteins into areas that will eventually form functionally distinct regions. And so what we can learn from this is that just a few molecular gradients can create substantial diversity within a space. And we can also understand that that space itself is involved in functional organization. But what about more complex examples like, say, the human brain? Well, we know that there are certain proteins that are organized in a gradient-like pattern throughout the brain. And in fact, molecular gradients have been hypothesized that span the different dimensions of the brain and may drive functional organization starting in very early development. So here I've made them a bit easier to see, and there are hypothetical gradients that traverse individual compartments vertically, that traverse the entire developing brain longitudinally, as well as horizontal gradients hypothesized in the cerebral cortex. But these gradients are incredibly hard to measure because it's very rare to have high throughput omic data with sufficient spatial sampling in human brains, especially in developing human brains. So we still know very little about how these gradients are involved in human brain organization. Now we do have one data set where we do have extensive spatial sampling of the brain, and that's everyone's favorite data set, the Allen Human Brain Atlas. So that's, of course, 8 hemispheres from 6 donors with combining for 3,700 tissue samples, each with expression data for 15,000 genes after quality control. Of course, these are all adult human brains. So this brings the question, would we be able to detect developmental molecular gradients, or at least their remnants, in the adult human brain? So that's what we set out to do in this study, to try to detect spatial gene expression gradients in the human brain. The way we decide to do that is a little unusual. What we're looking for are multigenic gene expression patterns that radiate linearly in one direction through the Euclidean space of the brain. So since we're literally looking for spatial autocorrelation, what we ended up doing was using a cross-decomposition model, sort of like partial least squares, to find patterns of gene expression that covary over space. So the two data sets that we cross-decomposed were gene expression at each sample as x, and the three-dimensional linear coordinates of that sample as y. So the resulting components would represent exactly what we're looking for, gene expression that covaries in a linear fashion over space. Importantly, we use data from the entire brain here, not just the cerebral cortex. So we divide the Allen data into training and test sets. In the training set, we found the 3-component PLS regression model works best, explains the most variance using cross-validation. And those 3 components each explain more variance than would be expected by chance. And we then used this PLS model to predict the 3D spatial location of left out samples in a test set based only on those 3 gene expression gradients. And we found that the model predicted location very well along the rostral, caudal, and dorsoventral axes. And, not quite as good, but good in the medial lateral axis. So certainly a solid validation of the model. And just to get a sense of what the brain looks like when predicted using these 3 spatial gradients, well, here are the actual y and z coordinates of all of the samples in the brain from the Allen Brain Atlas. And here is the predicted coordinates based on our PLS model. The overall average in our predictions was about 12 millimeters. And visualized here is that error distribution. You can see, unsurprisingly, the model seems to do worst at some of the spatial extremes of the brain, namely in superior parietal and prefrontal cortex. All right, so that's all well and good, but in the past, people have raised concerns that the autocorrelation we're measuring here might just be something study-specific, perhaps due to batch effects from the way that the Allen data was processed. So we wanted to see how well this model generalizes to other data. We found 3 other data sets with bulk regional gene expression data from adult brains, but the problem is that these data sets don't have that high spatial sampling and they don't have accompanying coordinates for the tissue samples. So we used the PLS model trained in the Allen dataset to transform the gene expression into components. And then we could see the degree to which the expression of those components across brain regions correlates with the regional expression in the Allen dataset. And we're showing that here. So we found really remarkable regional expression of the components across data sets. And this means that we can be sure that the genes contributing to these components show highly similar regional expression across these studies. So we can be sure that we're comparing apples to apples when we look across data sets. We'll come back to this later. So now that we feel good about the brain gradients generalizing, let's have a look at them. So here is the correlation I showed you earlier, representing the latent covariation between genes in space. And here are those latent gene expression and spatial gradients projected onto the brain. So the set of brains on the right here are the genetic gradients, which literally represent variation of a set of genes across space. So it's not surprising that the transcriptomic gradients on the right resemble the spatial gradients on the left, because that's by design. But what's more exciting is that despite this being a completely unsupervised approach, the expression gradients beautifully match those 3 hypothesized expression gradients from the literature. So here you can see clearly this first component, which moves from dorsal to ventral aspects of the brain, the second component that spans the anterior-posterior axis, and the third component which shows a sort of dorsolateral to medioventral pattern in the cerebral cortex and looks a lot like those horizontal gradients. So now let's get back to that question that we initially asked in the introduction. Does the confluence of these 3 whole brain gradients contribute in any way to the regionalization of the cortex or its functional organization? And so here we project the 3 transcriptomic gradients onto the cerebral cortex, shown here. And just like in the cartoon in the top left, we want to see if the overlap of those gradients forms regions of functional significance. So to figure out their overlap, we perform a hierarchical clustering to find meaningful territories of gradient overlap. And we see that multiple unsupervised cluster metrics pretty definitively suggest a 6-cluster solution. When we visualize those territories, well, surprise, they look pretty much like the lobes of the brain. You can see distinct regions involved in motor behaviors in blue, visual behaviors in yellow, spatial cognition in purple in those parietal lobes, language function in the turquoise temporal structure, the pink area encompassing subcortex, hippocampus, and insula involved in things like reward, anticipation, encoding, and pain. The prefrontal green structure is especially involved in social cognition. These are pretty much the major functional territories of the brain. And this really emphasizes how these distinct functional structures are pretty much maximally separated in Euclidean space. So, star graduate student in our lab, Valerie Sidner, published this beautiful review in Neuron describing the sensory-motor association axis of the brain, which is just such a spectacular piece. It's worth a read for everyone if you haven't already. But it outlines a very common pattern in the brain that seems to be the backbone of cortical variation across many, many different features. And so we wondered how our transcriptomic gradients here would stack up against the so-called principal gradient in explaining features of the brain. And to make it fair, we compared our 3 omics gradients to the first 3 functional gradients. So for each of the brain features you see in the figure to the left, we fit linear models to see which set of gradients better explains variation of the feature across the cortex. And we see that across all features, the gene gradients explain more variance. And so this provides further evidence that these gradients combine to create distinct structures. But hey, why compete when we can work together? We next fit models using both sets of gradients to see whether these 2 sets were contributing non-redundant information. And sure enough, this was the case for several features indicated here with white stars. And I'd like to draw your attention particularly to aerobic glycolysis in the center. The fMRI gradients and the transcriptomic gradient together explain 75% of the spatial distribution of aerobic glycolysis in the cortex. So it's very interesting to think about how these two organizational elements interact as it relates to brain energy usage. As a last set of analyses, we wanted to know whether these brain-wide transcriptomic gradients are consistent across species or if they're unique to humans. We were also curious whether we would be able to detect them in fetal brains to determine whether these were actually fetal gradients or instead just the remnants or the detritus of them. So I take you back to this slide where we use an approach to look at how well components generalize across data sets, and we can actually do this at the single subject level where we can, for each subject, determine how well the subject's regional gradient expression correlates with regional adult expression in the Allen data set. So we did this approach across 6 adult human donors, 5 adult chimpanzee donors, and 5 adult macaque donors, and we found that within subject regional correlations with the Allen dataset were really quite high across all 3 species, particularly for the first 2 components. So in other words, the expression of these 2 components was highly reliable across individuals of different primate species. We did find a component by species interaction where the third gradient, which showed the highest degree of variation within the cerebral cortex, I'll remind you, was less consistent in the 2 non-human primates. And so what about the development of the gradients over the lifespan? Well, once again, we calculated regional gradient expression across individuals, this time across different ages from 8 post-conception weeks to 40 postnatal years. And again, the values on the y-axis represent similarity to adult expression. And as you can see here, the gradients have different developmental timelines, with the first component reaching adult-like levels faster, while the other 2 components have an accelerated development that finally reaches adult-like patterns in, well, adulthood. What's more is that we were able to validate these timelines in another lifespan dataset of humans, and they actually also replicated in macaques as well. So this is a remarkably consistent finding. But it does mean that the components are not fetal gradients, except maybe component 1. But it suggests that the gene patterns themselves develop in accordance with gradient-like spatial organization throughout development, and they don't really converge until adulthood, which is interesting. So in summary, we identified patterns of gene expression that occurred systematically along spatial gradients in the brain. The components were reproducible across and within cohorts, likely contribute to cortical arealization. These gradients are rather consistent across species, and at least the adult form of them converges toward the end of brain development. And with that, I'd like to thank my talented and brilliant co-authors, especially Jacob Seilitz, who spent many hours on Zoom with me imparting his vast knowledge, and of course Ted Satterthwaite for leading the project. And I'd also like to thank our funding bodies, the Open Access Public Datasets, for making these analyses possible, and thank you for watching this video.